been given this title by Piero, which was really to try and cover both diagnostic and therapeutic approaches to optic nerve disease. I have to say that, bearing in mind we are running already behind time, it will probably end up just concentrating on the diagnostic advances, which is probably just as well, bearing in mind our last two cases have very much been on that. So in medicine as a whole, there is a new idea that when we talk about diagnostics, we should also be considering the concept of biomarkers. So what do we mean by biomarker? This is the official definition. It is a characteristic that is objectively measured and evaluated as an indicator of normal biologic processes, pathogenic processes, or pharmacological responses to a therapeutic intervention. So in other words, it doesn't necessarily only indicate the person has disease, but it may lead to predicting how they will behave in the natural history of that disease, and indeed how they might respond to therapy. So this has come about because there's been a real need in specialities like cancer that you have surrogate markers to tell you whether a treatment is going to work or not. So you're not wasting time in giving your patient something which actually is probably not going to work. The best demonstration of this would be in the world of Alzheimer's disease. And I'm sure a lot of you are aware that I think it was yesterday in Nature, there was another breakthrough that there are two groups that have got together and worked out that you can actually use fragments of the plaque protein in the brain, beta amyloid. You can actually detect small amounts of these molecules in the blood as a marker of disease activity. And this all comes from this huge collaboration that's been going on in Alzheimer's now since around 2000. And this was the first time you had academics, clinicians, and industry getting together to try and tackle the problem as to whether we could identify new biomarkers for this disease, Alzheimer's, which could be used to then assess therapy. You may have realized now that they've not done terribly well, which is one of the reasons why, for example, Pfizer very recently has pulled out of doing any further work with Alzheimer drugs. But the key is, because of this collaboration, within a matter of 5 years, several things emerged. One was that you could use the levels of beta amyloid in the cerebrospinal fluid as an indicator of disease. The other was you could use the scanning hippocampal volume, and also you could use tau and a natural history of the changes. So, for example, beta amyloid in the CSF goes down first because it's believed to be deposited. This is believed to be an early indicator. The problem is that when you're talking about treating a disease like Alzheimer's, actually you tend to only treat it around here. So probably the use of the early biomarkers hasn't quite gone through, but it is an example of where we are going in diseases such as this. So what have we got in optic nerve degeneration? Well, you've heard a lot about neuro-ophthalmology today. I'm going to concentrate a little bit more on glaucoma. And I think you'll agree, at the end of the day, we've really only got 3 options in trying to assess disease. We've moved on from this marker here, intraocular pressure. We've got to visual function, and I think that is still our gold standard. But increasingly, and especially with Piero's influence, the idea of structural changes being acceptable markers of disease is coming through. So our question here is, do these adequately have evidence that they could be used as biomarkers. One of the ways one is supposed to test if something is a biomarker is to answer 3 questions. One is, can we say the analysis of that biomarker is effective? Secondly, can we associate it with a clinical endpoint? So usually in glaucoma, we mean vision loss. And finally, can you use this realistically in our everyday practice? So something like perhaps electrophysiology has run out of favor because it takes such a long time for the patients to do, and the sensitivity rate may not be very great, that it may not fulfill a utility type perspective. So where are we with visual function? I think Anders Hale has a lot to be thanked for in bringing across the idea of rate of progression. So you remember with this, what we're talking about here is you're using a global parameter, the visual field index, to work out how your patient is going to progress over time. So in this 57-year-old person with a mean deviation of -5, which at that stage was called early. I think we could argue whether that really is early disease now. If you had successful treatment, and here what he's suggesting is even a reduction of 2 millimeters of mercury, you could alter the steep incline of your rate of progression to prolong the deterioration, so it's actually less, so it takes a 7-year delay. Okay, so that would fulfill what one would call evidence that rate of progression for visual fields could be used as a biomarker. What about OCT? I'm not going to go into a lot of depth here because you've heard so much about this already, but these are the accepted and validated indices that are currently used. These are coming through Brooks membrane opening, the macular changes, which we've already heard about, posterior pole analysis, and of course the ganglion cell complex type measures. So all of these are potentially very important biomarkers for the future. One of the things that we have to bear in mind, though, is if you remember one of the slides I showed you further back, is you have to show that there is an influence on your final endpoint. And the final endpoint in regulatory terms, and I mean particularly the FDA and Wiley Chambers, is visual function. And so ultimately at the moment we're left with having to link the structure with function. And there are a number of these things that have come about. You can use the rate of progression in addition to just visual fields. You can use it even in OCT. Okay, and the rate of progression in OCT is now part of most of the readouts that you get automatically in the software with whatever OCT machine you are given. And with respect to it being a surrogate, when people have looked at the correlation of whether your OCT is going to predict a change in your visual field, there is very good evidence now. For example, if you look at the average superior retinal nerve fiber layer thickness, you can predict, as you can see here in pre-parametric glaucoma, that you're going to end up getting a visual field deterioration. This recent paper is probably being put forward by Zeiss for one reason why OCT should be considered as a biomarker of glaucoma. You can't see exactly the text here, but really what it's trying to show is this is when this particular patient gets a visual field defect. Actually, if you look at the rate of progression with the OCT, it predicted it 2 years in advance. Okay? So it's a 2-year prediction of an endpoint, which would therefore suffice as a surrogate. And that's led to the evolution of this sort of work. Again, you've heard about that already. All of this is suggesting that if you combine OCT with visual fields and you get good correlations, that may be a good indicator of health. But what about the future? And so there are a number of structural markers that are coming through. OCT angiography we haven't really heard about today, and that's because it's still not validated. But it may be very promising, especially if it is going to help us predict who is going to get worsening disease. And if you look at the segmentation that's going on, looking at the different layers of the eye and what OCT angiography and blood supply can provide, that may be something exciting in the future. The other one is OCT-AO. So looking at the lamina corbosa itself, we've been doing that as clinicians for a very long time. But look at the resolution you get with adaptive optics, okay? And one of the nice things about that is if you can quantify that and follow it up over time, not only is the change in each of these lamina corbosa pores important, but also the importance that you can follow it up longitudinally. So I think you're probably going to hear a lot more about this as we come to terms of it. The last one is dark, and I think the last time I was in Italy, we hadn't quite reported these results because it took us a jolly long time. So for those of you still unfamiliar with this technology, I'll just go into a little bit of detail about it. So DARC is the name given to the technology that we've developed at UCL and at Imperial, which stands for the detection of apoptosing retinal cells. And it's based on a very simple process, which is the fact that sick cells, instead of having these molecules called phosphatidylserine, which normally live on the inner membrane, when the cell becomes sick, those molecules go to the outer membrane of the cell, here, and annexin 5 is a protein that is endogenous, we all have it, that has a high affinity to these phosphatidylserine molecules on the outer side of the cell. And so this works like an indicator that your cell is stressed. So what we proposed many years ago now, 2004 in this paper, was that if you fluorescently label your annexin, you can actually at a cellular level pick up individual retinal ganglion cells apoptosing. So these white spots you see here is a video actually in a rat eye where we showed that you could detect apoptosis. And we say at the end of that paper that actually it won't be long before we translate it to the patient. And Piero and Chiara will remember me saying that when we probably first met a few years ago. It's taken longer than I would have liked, but it's led to a very nice collaboration, unique collaboration between UCL, Imperial, and the Wellcome Trust. So we've now finished phase 2. In fact, we finished it in July, and the database has been locked, so we should be able to give you the results, I'm hoping, within the next month or two. So the phase 1 paper was published in April last year, and we hope to have the publication of the phase 2. I'll go through phase 1 with you today, but we still have to report on our findings in addition to glaucoma, to macular degeneration, optic neuritis, and we used, as a model of Alzheimer's disease, some Downs patients. So the phase 1, this is a diagnostic test that actually involves an intravenous injection. So because it was being given for the first time with a fluorescent marker anywhere in the human being, we had to go through a proper regulatory pathway. So this is a proper phase 1 where the primary objective had to be safety and tolerability. But we did have a secondary objective because in addition to using healthy volunteers, we were also able to use glaucoma patients. And so we were able to look at what we've called the dark count, which is the number of apoptosing cells that are present in the retina, and compared those in normals to glaucoma. So it was on 16 subjects, randomized between them to different doses of the intravenous agent, annexin 776. 8 of these were early glaucomatous patients, and 8 were normal healthy controls. So remember, it was primarily for safety, and pharmacology, but we were also able to image those patients repeatedly over a period of 6 hours after they had the injection. Now, for glaucoma, what we had to we basically included in this trial were patients who were defined as progressive by any parameter that we had available. So that included visual fields, HRT, and OCT. So they had to show progression, which was statistically significant as a rate of progression in at least one of these parameters. So this is a breakdown of the baseline characteristics. This is a video of the actual investigation. This is Eduardo Normando, Italian from Salerno. As I said, half of my research group are Italian, so we do very well from Italy. You can see this is a very simple investigation, very similar to a fluorescein angiogram or an ICGA. You give the injection and capture in real time the images on the scanning laser ophthalmoscope using the fluorescent setting for ICG. So the marker that we've put in here is fluorescent at the near-infrared wavelength, very similar to ICG. So here you can see the results. So concentrate on this picture here, which is the raw image. This is the timeline. Over here are highlighted in yellow circles where the spots are appearing. And this is the final picture. So I'll go back so you can see it again without having to look at all those things again. So as I said, keep looking over here, and if you watch carefully, I'm not sure the lighting's good enough, you'll begin to see these spots appearing over a period of 1 hour, moving on to 2 hours, 4 hours, and finally 6 hours. So you can see here, these white dots are representative of single labeled cells which are possibly apoptosing or sick, but we wanted to see whether this was an indicator of disease. So firstly what we did was we looked at the dark counts in patients who had glaucoma compared to those who were healthy. So remember, this was a very small group of patients, but we still showed that there was a significant increase in the level of apoptosis in those patients with glaucoma compared to those healthy controls over there. But what was more fascinating, because remember this is a very small group of patients, was when we went back and we repeated the gold standard tests, so all those baseline glaucoma tests that I showed you, we kept on repeating right up until 18 months after they'd had their single injection of annexin 776. And you can see what we showed was actually those with a higher dark count went on to progress, suggesting this could be a surrogate. So the summary of that was that actually this was the first time that anyone had visualized single cell apoptosis anywhere in the body in a human trial. And there was an increased level of activity in disease, but more interestingly was that this effect of a surrogate may be present, which is that it was predictive of an increasing rate of progression by gold standard machines. So as I said, we've moved on to phase 2, and we should hopefully start a phase 2B soon. Our phase 2A includes macular degeneration, Down's patients as a model of Alzheimer's, and optic neuritis patients. And our idea here is one of the things that has been coming through, and maybe one of the areas which has been a little bit stagnant recently, is for neuroprotection, you cannot really follow effective therapies or actually look to see if they really are effective, hence memantine or even the Logix trial, without having a very good way of monitoring that and looking at that. And DARC, as well as all these advances in OCT and AO, I think may allow us to do that in a much shorter time scale than was possible. I'm not going to go into the neuroprotective side, but I'll just end with just a summary of what I presented with phase 1. As I've often said, Italy is our hotbed for neuroprotection, And I'm looking forward very soon, hopefully, to do multi-center trials with Italian colleagues here, where we'll be using dark, hopefully, as a marker of disease and treatment response. I think treatment response is going to be very importantly. And this work really has been possible due to a large number of people, a lot of them Italians, and particularly the Wellcome Trust, who gave us £4 million to allow us to take it to the patient. So I'm very happy to answer questions now. Thank you.